So we were done till the basis intro. We were discussing nucleotide. So here, oh god, sorry. Here you can see this was nucleotide. So nucleotide is consist of three parts. It was phosphate, nitrogenous bases, and sugar. So here we have done the bases, nitrogen bases. These two parts are remaining. So let's start with this in this video. So we are discussing that is sugar. Okay, so sugar, which is like beta sugar. In this you will study beta sugar. So if I have written any one, this means there should be a concept of alpha as well, or what would be the difference. So I'm just going to tell you this is the structure. So here you will see that OH is present on the upper side and H is present on the upper side, downward side. OH here, H here. We have studied this in the carbohydrate in very detail. So I'm just giving you an overview of beta sugar. Beta sugar is simply when H is present on the downward side, that is known as beta sugar. Why I'm just saying that? Because in other structures you will see OH will be present. Like here, OH, when OH will be present, this means that is alpha sugar. That is alpha. But when you see only hydrogen is there, this means that's known as beta sugar. So in these sugar, ribose sugar, like 5 carbon containing sugar, you will see the structure that will be of beta sugar, okay, not alpha. So here you will see another structure. I'm making a structure, then I will just show you, or let me just have with a good color. So here you will see like this. This kind of a structure. Here you will see so here OH will be present on the upper side as I just told you, then downward there will be H on, but here I'm going to discuss another case like exception. Here you will see OH, here will be CH2OH. This is a basic structure we have done this in very detail in previous videos. So this is the structure. Okay, so what I was just telling you is that here OH is present. This means this is alpha sugar. Okay, and we have done the difference between RNA and DNA. So when you see on the second sugar there is change. When you see only H is present or only OH is present. So when H is present, that means that is forming RNA, because DNA means deoxy, D means removal. So when you see O is not present, sorry, this is DNA because O is not present, OH, this is RNA. This means here is the removal of O. So here you see this only H, and here's only OH, here's H on the second carbon. When you see only H is present, this means we can say the change is only possible when you have beta 2 sugar, and that will form the structure of deoxyribose sugar. Deoxyribose sugar.
Yeah.
Okay, this is sugar, and secondly this is deoxyribose because there is exchangement deoxyribose. And one more thing, let me just move down. There's one concept of base. Base is a structure, like base is also known as heterocyclic. Heterocyclic, which means when hetero means different, so different rings combined they form hetero. So this was the remaining part of the base. We are done with the base sugar. Now next video would be phosphate. So base hetero, which means different chain, when combined they form a structure like NN will be